In this lecture, I will discuss about the virtual screening of compounds to identify lead compounds from a pool of different ligands. So, in the earlier lecture we discussed about protein ligand interactions. What is the ligand? Small molecule which can trigger the activity of the proteins that they when they interact with the active site or the binding site. So, this can be activator or the inhibitors or neurotransmitters etc. right. So, when they interact right with the protein right. So, you can identify different types of inhibitors for any drug targets. So, what are the different types of docking did we discuss?
Rigid and flexible.
Rigid and flexible docking. What is rigid docking?
So, both parts.
So, you can see the binding site of the protein as rigid. Ligand also have one conformation you can dock, you can make different conformation for ligand, you can interact with the protein right to get the score. What is flexible docking?
There will be some.
So, here the proteins are also flexible. So, you can allow different degrees of freedom right. In this case you will get the different sampling. So, for this sampling you can get this function scoring function. So, you will find the best score to identify the target for any particular active site. So, to do the docking we need proteins mainly the active site and different poses right as well as the ligands right. Then you can do the types of docking. So, then we do the docking. What are two different aspects we need to consider?
Search algorithm.
One is a conformation sampling and the second one is scoring function right. What are different ways to sample the conformation of the protein at the active sites? Systematic or stochastic right. The systematic we need to systematically carry out all the possible conformations, it is time consuming right, but you can get the data, it is good for the small compounds. So, if you have the stochastic one, so in this case randomly it can choose right the different types of algorithms we discussed. Which algorithms we discussed for the sampling? Simulated annealing and the genetic algorithm right. So, we use different algorithms to sample the proteins conformation. Then regarding scoring function, so what is scoring function?
Which can score?
This is a kind of energy functions, so any function which can provide the binding affinity in terms of some values that are called scores. This will tell you for a different pose of any protein at the active site which ligand can fit right, the best with this particular pose. So, different types of scoring functions you can decide which ligand is the best right. For example, the shape or chemical complementarity, you can see empirical potentials, energy, then force field, then we discussed about the propensity values right, we calculate the propensity then convert it to the potentials right, knowledge based method and the consensus right, you can see the use of various combination of different potentials fine. So, there are several software available in the literature right, some of them are publicly available for example Autodoc and the Glide right, this is available as a part of Schrodinger right, you can use for the docking. So, now the question is docking is the only way to identify okay, which ligand can fit with this particular pose. So, if we have a pool of compounds for example 5 million compounds right, how to identify the potential lead compounds right. This is the technique called virtual screening. So, this is the computational or in silico algorithm for screening these compounds. So, what is the aim of the virtual screening? Okay, we have the proteins and the ligand, this will give a score and this can rank these ligands and also filter a set of structures whether this can fit or this is not fitting with any particular conformation in the protein site. So, you can use docking to do this right. So, in this case you can filter the compounds and you can and get some libraries to synthesize and you can also suggest to purchase some compounds for the experiments. In any case final experimental validation is required right, even if you identify any lead compounds using virtual screening the computational techniques. So, now if you give the protein here, so where is active site probably you can see this is fitting here. So, you can see some cavities and here there are many ligands, you can see database of ligands. So, what the virtual screening does? So, given a protein and database of ligands right, we can use several software right. So, see from among all these ligands which ligand will fit with this particular protein. In this case which one is the best fit? NAD, right. You can see the NAD is found to be best match at least for this combination, right, for this protein from this list of ligands. I show some more examples. These are data of the oxygen transport molecule, right, the surface and the myoglobin ligand, and here you can see influence of virus B, right, the neuraminidase complex with the xanamivir. This is xanamivir here, this is the protein site, how they fit, right, in the core. This is another one HIV protease. Here also you can see aspartyl group, this can be acting as these active sites, so that the ligands can go and interact, right, at these sites. So, there are several examples available in the literature, right, for the protein ligand complexes. So, then if you look into these available ligands, many structures you can directly reject based on some of these chemical structures as well as the availability of the rings or the Lipinski's rule of 5. For example, if I showed several rejected compounds based on various aspects. For example, take this one. So, here is a molecular weight is 837, right, hydrogen number acceptor is 15. So, these two violated the Lipinski's rule of 5. Right, what is the Lipinski's rule of 5?
Molecular weight it should be less than 500.
Less than 500.
Hydrogen bond acceptor should be less than 10.
Less than 10.
And donor should be less than 5.
Less than 5.
And log.
And the partition coefficient is less than 5, right. So, here if you see compound number 1, this compound molecular weight is 837 and hydrogen bond acceptor is 15. So, in this case it violated the two aspects. So, this may not be a drug like molecule. Then the here one is rejected mainly because it is insoluble in water, because solubility is important, because if it is not soluble, right, in this case if you cannot drink, it is not soluble, right, in this case it is rejected. So, here this one is having many rings. So, because of that they rejected this compound. Here you can see, sir, they depending upon the inorganic and the heteroatoms. So, in this because of the reason, okay, they rejected this compound. So, here there are several toxic groups, and here you can see several reactive groups, right, some cases multiple chiral centers. So, these are the various aspects how we rejected some molecules which may not be a drug like molecule. Then what do we need? What do we need in the virtual screening? So, there are set of compounds. So, there are several actives, some of them are inactives. So, we need to identify the actives or inactives and reject the inactives and get the positives. Then we have to get the quality in our lead, because it should be quality, then this can be potentially, it could be a drug. So, you got to reproduce the binding mode and predict the binding affinity and rank the diverse compounds, right. Then we get the hit rate by database mining, right. Then we have to reduce false positives and false negatives, right, and then it should be fast to get this structure based drug design. So, there is various steps you need to do for screening the compounds, right, from the pool of compounds to identify a lead like molecule. Okay, I show one example. Okay, for here you can see the CS kinase protein, right. This is involved in this colorectal cancer. So, the target is the CS kinase, right, how to identify the inhibitors for this particular protein. So, Drug Institute of Technology, right, in 2014 they organized a competition, right, because there are several methods available for docking, right. For example, we discussed about various software for AutoDock and Glide and many software. Likewise, there are hundreds of software available. Then different ways to get the probable compounds. So, it is very important to identify whether this any method can potentially identify the probable compounds. Second question is, are there any mismatches or matches or overlapping of the different techniques which can potentially identify same compounds, and whether they can get the diverse variety of compounds. If you have different methods and you get the diverse compounds, then you can reduce the pool of compounds to a limited set, and that you can go for the experimental validation. So, there is based on that background they organized a competition like the CASP. What is CASP? Critical assessment of prediction of protein structures, right. So, likewise here they listed up given up 2.2 million compounds from enamine. Why they used enamine compounds? Because all the compounds are readily available. So, do not have to synthesize and take time, right. So, then we can identify the inhibitors with known structures, and once we submitted the compound, then they will do experiments and then compare whether any of these compounds are potentially inhibiting the activity, right. This is the major role for this initiative of the parallel bioinformatics. So, like explain. So, what is CS kinase? Okay, you can see the CS kinase. Okay, this is the from the kinase family, right. So, here this is also involved in the colorectal cancer. So, it is one of the major target. It has several domains, for example, SH3 domain, it is 2 domains. These domains are similar to the other family members like SRC, S, FIN, line and so on. So, it is a catalytic site, right. Here you have the 2 phosphoryl sites, one is Y416, right, another one is Y527. Okay, here you can see this is SH3 domain, this is SH2 domain, and here this is the linkage, and here is activation loop. This loop is this part is very important for this inhibitor. So, if you look into the literature, so crystal structure of CSRC kinase is known, but CS kinase is not known, but the homology is very high with the other members. So, tyrosine 416, so it is in the activation loop, this is the activation loop here, right. So, here is the tyrosine 416, right. This undergo phosphorylation during the activation of kinase, right. Then how to identify the probable inhibitors? So, to have docking, right, we need the protein as well as ligand. Here the protein, what is the protein here? CS kinase. CS kinase is a protein. So, protein we know, but protein structure we do not know. Then the ligand, what is the library of ligands? Enamine library. So, the last class when we discussed about the docking, before docking first we need to prepare the protein as well as prepare the ligand, right. So, now we have the enamine library, we need to prepare the ligands and we have the protein. So, protein also we have to prepare, right. I will explain what we do. So, first we get the enamine library 2.2 million compounds, we need to optimize and do check all the parameters whether it is stable and this fine or not. Once we optimize, then we can calculate the physicochemical properties, the various physicochemical properties, for example, molecular weight, heteromer donors, acceptors and the molar refractivity, right. So, various physicochemical properties, you can calculate these properties. So, this is with the ligand. Then among these 2.2 million ligands, because several ligands we can reject, because I discussed about the compounds which you can reject at the beginning. So, we do not go for the screening and the final checking. So, we can reject based on the some of these characteristic features. So, for that purpose what we did, so we get the CS kinase inhibitors from this experimental data called the binding DB. This has some data for the CS kinase inhibitors, known inhibitors, right, but there is not available in this enamine library. If it is available in enamine library, they remove that one, right. So, now all the 460, then some of them are similar, some of them are diverse. So, we need to have the diverse compounds. So, get the diverse compounds using Tanimoto coefficient, I will explain soon. In this case, you can reduce this 460 into 159 ligands. Then among the 153, we calculate some of the physicochemical properties. These are called actives, because already known known binding affinity. So, they are known as inhibitors, right. So, now we compare these physicochemical properties, right, and the physicochemical properties of these 2.2 million ligands, we compare with the deviation, then you can remove several compounds. Finally, we ended up with only 5200 compounds. So, you can drastically be reduced from 2.2 million to 5000 compounds. Then we added some decays, because to see whether the decays are correctly eliminated and the actives are correctly identified. So, with this 5000 compounds, we added 159 actives and the 6000 decays, so that this will be 11,761 compounds. Fine, this is now the ligand side, we fixed this type of compounds. The protein side, so we know the target is CS kinase, structure is not known. So, we can have the template, they called the SRC kinase, which you can identify more than 90%. So, we can, if it is more than 90%, what can we do? We can do homology modeling, right. So, we can homology modeling, we can do homology model. So, we can do homology modeling of this CS kinase, but this will have only one static structure, right. In this case, we will get only one conformation, right. So, the protein also have different type, different conformation, not only the active sites, but other places also they have different conformations. Accordingly, the conformation active site also change. So, in this case we can do MD simulations, right, the simulation mainly this kinase domain, right, and take the different conformations and cluster the conformations, and finally we get some 7 structures. These 7, one is homology model and one is the SRC kinase. Take this as proteins. Then with these ligands and these proteins we can do docking, because now we significantly reduced the ligands. We can use the high throughput virtual screening or simple precision or the extra precision. We will do all these screening methods to get the final compounds, right, say 120 compounds, right. Okay. So, first we prepare the ligand and compare the ligand with the known actives, and finally we reduce the total number of ligands based on the physicochemical properties. Then included the actives and decoys for the test purpose. Then we prepare the protein side using the homology modeling and the MD simulations, and we make the docking, and finally we get the compounds and we can screen it, right, fine. So, how to do this? First we get the ligands. So, NML we get 2.2 million compounds, right. Then to see whether these compounds have any similarity or how we can identify the inhibitors for the CS kinase, we check the binding DB. So, in this case we have the actives already reported, like the 1 to 1000 nanomolar range. So, here you can see 460 compounds. Then due 6, this will give you the number of decays. These are known non-binders for the SRC family. This is a negative deltas, right. So, we got the negative, we got the positive, we can use this as a control, and based on that we can identify the new inhibitors, and we can thus validate using experiments, right. Now, first we get the ligands, first we do the optimization, right. We discussed earlier, like we need the stereoisomers and the different tautomers, and we can do the ionization and so on, right, make all these things perfect, and we generate the coordinates using all these various possible states. We generate different coordinates. Then you minimize the structure using the OPLS force field, that is a kind of force field you can calculate the energy, right, and finally retain the lowest minimum structure. For each ligand we try to get different conformations based on the possible states, and we generate the coordinates and minimize the structure, we retain the minimized structure, right. Likewise we can generate, right, prepare the ligands. Once the ligands is prepared, then we can use the ligand for calculating the physicochemical properties. For example, in this program quick probe, they calculate more than 80 physicochemical properties. For example, molecular weight, dipole moment of the molecule, and acceptable surface area, and the hydrophobic component of this area, hydrophilic component and pi component, and so on. So, they can calculate various properties, more than 80 properties, and among the different properties we can select the best properties which can fit or which are important for binding, right. What are the various properties which are mainly important for binding?
Shape complementarity.
Right, you can see the hydrogen bonds, right, then you can see the log P, you can see molecular weight, some properties are very important, right, along with some other properties. You can take 5 or more, right, depending upon the properties which are important for the binding with the target, right. So, now they filter the ligands. For example, if you have the ligands 1 and 2, right, either you take both or you take only one. If you have the sequence non-redundant sequence structures, sequence identity, how to get the non-redundant sequences?
Cluster.
A cluster, we get the cluster and you can see similarity, we take the similarity of 40% or 30%, right. Even they are similar, then maybe you can take one, right. If they are not similar, take both. This is non-redundant sequences, right, you can make. Like with the ligands, if you see there are different types of elements. Say ring is present in first compound, yes, right, ring is present in second compound, yes, then the second one is this OHO is present here,
No.
it is here, right, OHO the same, right, this yes is here, is here, right, this yes, then this one it is not available both. Then take this type of group, this O is here, right, here is here, yes, but here no, so put 0, right. So, you put one for any groups you put 1 if it is present, and if it is 0 if it is not present, right, 1 and 2. Now, you can see comparing these 1 and 2, number of bits set in both, how many cases you can see is available in both? 1, 2, 3, right, 3. So, B is the number of bits set in 1, but not in 2. How many cases present only in 1 and not in 2? 2. How many bits set only in 2, but not in 1? 0, right. If this is the case, you can calculate the Tanimoto coefficient using the equation A by A plus B plus C, right. So, A equal to 3, B equal to 2, C equal to 0. So, in this case this equal to 3 by 5, 3 plus 2, right, this 3 plus this equal to 60% or 0.6, right. So, they take the 460 compounds and compare the structures of all the active compounds, right, using the Tanimoto coefficient and set the cutoff of 0.5. If you do this, we ended up with the 159 compounds from this 460, right. Like the sequence analysis, structural analysis we use RMSD, sequence analysis we look the similarity of the similarity matrix, and here also we use the Tanimoto coefficient to get the non-redundant structures. So, from this 159 you can calculate the physicochemical properties, right, different properties you can calculate. For example, we take the 5 important ones, molecular weight is 450 plus or minus 75, right, in this case 375 to 525, log P is 3 plus or minus 2, hydrogen mode 1 has 2 plus or minus 1, acceptors 5 plus or minus 1, and the molar refractivity 120 plus or minus 10. So, we use both these ranges, right, minus and plus, and they screen the initial 2.2 million compounds. Finally, we ended up with these 5000 plus compounds, right. Then we have the actives 159, 6000 decays, finally we get 11,000 compounds, right, that is fine. So, then we need, this is the ligand side, that is fine, we like set with the ligands.